Hi everyone, thank you for joining our R for Bioinfo series that we started in September 2020. Today we have the great pleasure to have with us Dr. Janani Ravi. Thank you, Dr. Janani, for accepting our invitation. It's a great pleasure to have you with us today. So, Dr. Janani Ravi is currently an assistant professor at the Department of Pathobiology and Diagnostic Investigation, Michigan State University. She completed her PhD in computational biology at Virginia Tech and her postdoctoral research at the Public Health Research Institute, Rutgers Biomedical and Health Sciences. Her research group at MSU works on developing computational approaches to study the molecular basis of pathogenesis and intervention of infectious diseases using protein sequence structure function relationships, comparative genomics, and drug repurposing. Dr. Janani is also actively engaged in training, education, and outreach, focusing on increasing the participation of underrepresented minorities in data science and R programming. She is the founder of Our Ladies East Lansing and co-founder of Women Plus Data Science platforms that together engage more than 1,000 members. Thank you, Dr. Janani. So I will be the host of today's session. I'm Hediat Nani. I'm postdoc at Pasteur Institute of Tunis. Thank you very much. The screen is yours.
Okay. Thank you so much, Hedia, for inviting me here. It's a great pleasure. It's the first time I'm actually talking to Our Ladies Tunis or Our Ladies Africa groups. So it's my pleasure. And thanks for your kind introduction. So before we get started, I just want to make sure that everyone can see me, hear me, see my presentation. All good? I see some nods. Okay.
Everything is
in
okay.
the chat. Okay, thank you. So the link is in the chat for the GitHub repository. And maybe I can just very quickly get started there before we move on with our presentation. Okay. So if you click on the link, this is where you'll go. Okay, maybe it might not look black for you. That's just my favorite thing. Everything is black on my screen, but this is your GitHub repository.
Sorry, can you make the screen a little bit bigger because it's quite small?
Yes.
Yeah, good now. Thank you.
Better?
Yeah, quite better.
Okay. So this is the workshop material. And all of this has to do with Tidyverse, pretty much. So today I'll be talking to you about how to use genomics data because this is part of the R for Bio series. But if you're not from a biology background and you still want to play around with Tidyverse, you can check the other data set here, which is the Gapminder data set. And all the material, the rmarkdown, HTML, slides, all of them are present in this repository. Okay. Now, ideally, because it's a 3-hour workshop, I would like for you all to test things and work with these things along with me. Okay. So this is supposed to be a hands-on workshop. It might be a little difficult because it's remote, but I will try to help you as much as I can. Okay. So for the first thing, it means you need to have your code available locally. And for this, I would recommend one of two things. If you're familiar with GitHub, and if you already work with GitHub regularly, then I will suggest that you clone the repository, make a local copy of it, and then work with it. Okay. Now, if you're not familiar with GitHub, then what I will recommend is you just download the zipped file here. So regardless of which platform you're using, Mac, Windows, Linux, you can download the zipped file here and then open it up locally. And if you open it locally, you should see something like this. You should see the same file architecture pretty much. You should see the Genomics Gapminder. And what I would recommend is that you click on this R project file so that it opens in RStudio. Okay. And I hope you have Tidyverse installed. If not, I would recommend that you install Tidyverse because, well, we'll be using that today. So these are the packages to be installed. Install Tidyverse and install here. Okay. So one quick question that I have for you all is how many of you have used Tidyverse before? And how many of you have used rmarkdown before?
Please
You can
raise your
hands.
hand. Yeah, thank you.
You can also use the raise hand feature in Google Meet in case your video is off. Okay. So we at least have a third of people who have used Tidyverse. Now, can you raise your hands if you are familiar with rmarkdown? Okay, again, we have about a third of people. All right. Anyway, rmarkdown, that's also part of Tidyverse, so I can also take you through that today. With that, let's get started. So basically today, because we are doing this remotely, I would recommend at any point of time, if you have any questions, please raise your hand. If I don't see the chat, or if I don't see who's raising their hands, then probably Hedia or Mona can help me with that.
Yes,
Okay?
for sure.
And please feel free to interrupt me, because if you're stuck somewhere early on, I don't want you to be holding on to a question until the very end, because you need to understand the initial parts to understand the later parts. Okay, and there will be, after each section, there will be some time for you to try things out, and midway we'll probably have a 5 to 10 minute break. Again, I'm happy to take questions during that time. Okay, so that's the overall structure of today's workshop. And today we'll be talking about a beautiful suite, a package called Tidyverse, which is actually a suite of other packages, which includes packages for import and export of data with Reader, tidying up your data with Tidier, manipulating and wrangling your data with dplyr and Magitor, data visualization with Ggplot, and rmarkdown, which is amazing for reproducible research. Now, I have not mentioned Broom, linear models, and ggmap because, well, we only have 3 hours and we have to start somewhere. So on rmarkdown, we won't explicitly be talking about rmarkdown, but all the code is available in rmarkdown. So if you're able to understand what is there and how to knit it, then you've already learned a fair amount of rmarkdown by the end of this workshop. And these are the 5 different parts that we'll be covering today. Okay. So from all the raised hands, I assume that all of you have R and RStudio already installed. If you're using the native theme, it's going to look white and bright, unlike mine. And I hope you have installed Tidyverse. If not, you just use install.packages and Tidyverse within quotes. And that's the way for you to install your package. So especially if you're new to R, the way to think about installing packages is you install packages once, which is similar to installing, well, any application, but you load it again and again. For each session, every time you start to use something, you load it again. And the way you load it is by using the library command. And you can also see it visually if you go to the Packages tab here in your RStudio. If the box is checked, it means that that package is loaded. If it is not checked, it means it's installed, but it's not loaded. So a visual way to do it is to check or uncheck the box. But because we are going to do everything within a code, we are going to use the library to load the package. Okay, so every session, if you basically don't load the packages, you're not going to be able to use the functions that are within the package. So the very first step is data. For any analysis that you're going to do, you'll either use your own data or you'll use existing preloaded data sets. So there are many different ways in which you can load data. So these three functions here that I'm showing are read underscore CSV is to read any comma separated value file. So if any of you have used Excel or Google spreadsheet or numbers or any of these spreadsheet formats, you can basically export your data as comma separated values. And that comma separated value CSV file can be read in by R using read underscore CSV. And for each of these functions that I'm showing, there are going to be multiple arguments, but I'll just show you the ones that we most frequently use. Okay, and if you want to load the data set along with the column names, you just say yes. The first row is the column names, and I want the column names in my imported data. That's what this means here. Read underscore delim is a more generalized version of read underscore CSV, because CSV only works for comma. CSV2 works for semicolon. TSV works for tab separated. But let's say I've decided to import some really weird data set where they're using double slash or a hash or some other sign to actually separate their data. Right? In that case, I can use a more generalized version called read underscore delim, and I can specify what is the delimiter that's separating these different columns. Now, if you have used base R, you may be familiar with the counterparts, which is read.csv, read.delim. And most of the tidyverse functions have the verb and the actual thing that they're doing separated by an underscore. Most of the tidy functions look that way. And more recently, in the past few years, Jenny Bryan developed this beautiful package to bring all of them who are using Excel into R. And the way she did it is by building this read Excel package. So now you don't even have to convert your Excel file to CSV, especially if you have a multi-sheet, really complicated looking Excel file. You can just give the path to your file. You tell which sheet you're interested in and what is the cell range that you're interested in importing. So it's as simple as that. And within that cell range, if you have column names, you specify that, yes, the first column does have column names. Otherwise, I just make this F or false. Okay, so these things are not going to work for you because these paths are all fake paths. The file names are all fake files. But we'll look at some real examples next. And today, because you're part of the R for Bio series, we are going to be working with Genomics dataset. So many of you may be prepared for this workshop, which means you're already familiar with biology. So I'm not going to ask who is familiar with biology. I'm going to ask the opposite question. Is there anyone here who has no clue about biology or bioinformatics or genomics? So if you know nothing about these data sets, please put up your hand. Everyone else, please don't raise your hand. Not bad. There are only 5 people who say, I have no clue what these bio data sets are doing. Don't worry about it. You can just think of it as one other data set that you work with. All right. So let me briefly introduce the data set to you, especially those who are unfamiliar with genomic data sets or biology. Feel free to stop me with questions. So we'll be looking at a more or less typical RNA-seq dataset, which is the result of RNA sequencing. These are human RNA-seq datasets that we'll be using today. And this is the structure of the data. So experimentally, there are different technologies that were used to get this data, one or two. I'll tell you what those are in just a bit. So these are the different sequencing technologies. There are 19 different samples that were used for this experiment, and we have data from 6 human tissue types: colon, heart, kidney, liver, lung, and stomach. Okay. The nice thing about this data set, the reason why I've picked a slightly complicated data set, but a typical genomics data set, is because it has numerical values that we can work with. It has several categorical variables, and more importantly, it has a good amount of metadata that we can work with. So this is, in some sense, a perfect data set to use with Tidyverse to see how to do the initial data exploration, data wrangling, and finally data visualization. So now I'm going to go back to RStudio. So for those of you who joined after I introduced this, start here, start with the GitHub repository, either clone it or pull it down with the download zip, unzip it, open it up, open the R project file. And if you open the R project file, this is where you will be. Is the font size okay, or let me make it bigger? Is this readable?
It can be bigger a little bit more.
Little bigger? Okay.
Yeah. For me now it's okay. Now? For me it's okay, but
all right. So I'm going slow because I'm hoping that all of you are opening this R project file and you're opening, go into the genomics folder and open the workshop Tidyverse Genomics, which is this R markdown file here. Don't worry about how it looks. I've spent some time writing it, so it may look fully populated, but that's for your benefit. But the really cool thing about this R markdown file is I've exported it as a HTML for you. So the HTML is also available in the same folder. So what you can do is click on the HTML file and say view in web browser. And it's going to open up this way for you. Okay, so this is the exact content of what is in my RStudio. Okay. Let me also share this link with you via chat. So you can also have this open in the browser. So you can see how it looks once you implement it or once you knit the file. Okay. So we were looking at the reader part, right? So you will see what are the different packages. You've loaded Tidyverse. And these are the initial functions that I just showed to you. Read underscore CSV, read underscore DLM. And now we will get to our data set. Everyone with me so far? Okay. So basically what we'll be doing here is, let me open the outline. You've installed, you've done all of this, you've loaded Tidyverse. Let's go to loading packages. Now the nice thing about opening up your R markdown file is you can load or run chunk by chunk. And for each chunk, you'll see a little play button, the green play button here. So for instance, if I run this, it's going to run whatever is within that R chunk. So anything that is within these triple ticks is R code. And here I'm saying evaluate it and show the piece of code in my HTML. Okay, so now it has loaded both of these libraries. Then I'm going to the next chunk. So if you go here and run the next chunk, so this is the URL from where I'm getting the data set. Okay, so what I'm specifying here is this is the variable that has the path to my data set, my genomic data set, and it's a tab separated file. Now let's see if we can actually look at this file. No, it's a .gz, never mind. So here you're saying read underscore TSV, and you're basically loading this here. So I can either run it line by line, and the shortcut for that is Command plus Enter if you're using a Mac, Control plus Enter if you're using Windows. If you're using Linux, well, someone will have to tell me what the shortcut is. I don't know. Or if you want to run the whole thing, you can select the whole thing also and do Command plus Enter or Control plus Enter. So now we have loaded this data set. Okay, and the way you know that it has loaded is because earlier I did not have this here, but now I have this new variable resulting from the file that I have just read. Okay, if I open it, this is how it will look. I'll tell you what the different columns are. I'll tell you everything. So don't worry about the really huge file that you've just loaded. Okay, but everyone has it now?
Janani, I'm sorry to interrupt. So I have
Yeah.
a quick question here. In case if we do
Yes.
not know whether the data which we are pulling, is it comma separated or a tab separated or something else? So is there any default option we should pick if we do not know the data?
I would say just open up your file and see what the delimiter is.
Okay, got it.
That would be the cleanest way, which is actually what I'm going to recommend next. So the first thing I will say for anyone, I mean, especially if you're starting a new analysis, is to not be afraid of opening your data set wherever you're opening it, but just open it, look at it, see how many rows there are, see what's the delimiter. That will tell you what's the very first step. How do you read it into R? Now, if you use your read underscore TSV or read underscore CSV, these are the most common. So comma and tab are the most common ones. If they are not working, and if everything is jamming into one column, you might want to see what is happening, right? So there is a read underscore lines. So if you just want to read the file into RStudio, no delimiter, just read it, open it, see how it looks. Then you know, okay, so this is my delimiter, and then you go back and read it the correct way. Okay, so these are the fake examples that I showed you that are not going to work. So I just said eval equals false. But let's go to our real data set. So here, now you've read your data set. It has loaded your file and it says this is how it looks. Okay. And the main things to know are that AAAF, you will see this often in the data set. For example, here, you're seeing AA colon, AA heart for the different organs, right? And then you're seeing BA liver. So what do these A and B and F mean? The second A, the second letter A or F is basically adult or fetal. Okay. And the first one is basically telling me the kind of technology that I'm using. It's either Agilent or Biochain or Origin. And the last part of it, which is after the underscore, is basically which tissue it is coming from. At this point, that's all you need to know. Rest of it, when you're exploring your data, I'll tell you a bit more. Okay. And as I said, I said look at your data, right? So what are the most common ways to look at your data? So if you're using base R, you may already be familiar with the structure function that shows you what your file looks like. So when you run structure and your data frame here, which is this, it looks this way. It says these are the dollar says it's a column. So it says you have your gene ID column, you have your chromosome, the start and end coordinates for each gene, because this is RNA-seq data, or actually each transcript in this case, and which strand in the chromosome, plus or minus strand that it belongs to, the length of each of these transcripts, and then which sample it's coming from. And we are seeing that it has 57,000, almost 58,000 rows and 25 variables or columns. Okay. But because we used Tidyverse function read underscore CSV, so yes, the rmarkdown file or the R files that you are asking for are in this repository that you basically have supposedly downloaded or cloned. Then you would have opened the R project file, you go to Genomics, and you see this rmarkdown file here. This is the rmarkdown file that I currently have open. If you want to see the rendered version with your local browser, you will be opening this HTML file. Okay? The presentation is also here. But for the initial part, when I'm explaining, I would still recommend that you look at my screen, or if you have multiple monitors, great. Throw me onto one screen, just have me talking, and then you try it on your RStudio in the other window. Okay? Otherwise, I'll give you, if you're just having a single screen, I'll give you time to switch back and forth. So let me very quickly tell you what are the different ways in which you can look at your data. I'll give you a couple of minutes to try it. Then we can move on to the next section. All right. So we used a tidy function called read underscore CSV, or TSV in this case. So the cool thing about using a Tidyverse function is it automatically reads it in a very nice format called Tibble. And it tells you everything in a much cleaner way. So here you had your data types listed separately. You had something else mentioned here as the data type, and then you had it spread out this way, correct? But now it gives me a quick snapshot when I just give the variable name. It just says these are the columns, at least the main columns that I can see within the screen, right? And then it gives me just the head, which is the top 10 rows. And the other stuff, it just describes here. So you can also use head. If you're familiar with any other programming language that is not R, head is still a very, very common function. The default probably in Unix is 10 rows, but here in R, the default is 6 rows. So you just see how the top 6 rows look like. Glimpse is again a tidy function. So it tells you the first few entries of each column. So this is a slightly cleaner way of using structure. It gives you what the data type is right away, and it shows you what the columns are and what the actual entries look like. So right away, you see that these are all numerical columns. These are categorical, which tells you which chromosome it has. This is a character variable again. Now, if you're more comfortable with actually looking at it in a visual way, then you will use view. So it actually opens it up in a GUI format in your RStudio. If you want to see just what the column names are, you use col names. If you want to know the number of rows, number of columns, you can also see that here. But let's say you want to extract it. This is how you do it. The number of rows, end row, the end col. And if you want to subset your data, saying I want only the first 5 rows and the first 5 samples, which is from 7 to 10, right? So I'm eliminating all of these. I'm saying I'm directly starting from the 7th column. So it gives you a neat little matrix that you have subsetted. And if you want to write this, you do the opposite of what you did with read. If you use read underscore TSV, you use write underscore TSV. If you want to save it instead as a comma separated, you just change this to CSV. Okay, let's just run the TSV first. So I have not specified that it should save it in Genomics, so it's saving it outside. So if I open this file, this is how it looks. It's tab separated. Now let's do a simple switch. Let's change it to CSV. So the only thing that has changed is now the delimiter is comma. So it is as simple as that. Okay. And the default is that it keeps the column names. That's why it has kept it. Okay. So if you are following along with me, well and good. If not, please take a minute to just check out these functions, look at your data, and come back to me with questions, if any. Can you give me just a second, please? Okay, so any questions on the data import and very initial data exploration part? Okay. If there are no raised hands, Marta, do you have a question? Or is this raised hand from the previous question? Okay, so you don't have a question. Great.
Ali is saying he got an error. So
Okay.
Might be like a technical error, just some package. Thank you.
Yeah, so I think the reason you have an error is because you have not installed the package first.
I have installed all the Tidyverse packages and only.
Yeah, so the first thing you should try is, yeah, okay, let's go back here to installation and setup.
You have to install the package here.
So here, can you see lines 89 and 90? You have to install Tidyverse, you have to install here.
Okay, thank you.
Because you can only load packages that you have installed before. So what it's complaining here is it does not find the package, so it's not able to load it. And by the way, here is a really cool package because now all of you are working working out of the same R project file, right? So you've opened the R project, which means when you go here to the top right of your R studio, you can see workshop Tidyverse. So now all of our relative paths, so if you say here, our relative path is always with respect to this master folder workshop Tidyverse, regardless of what comes before that. So for instance, if I had used get working directory or set working directory, and if I've given this path, do you think this would have worked for you? If I had said Janani Ravi GitHub Tidyverse, would this have worked for you? No. Right? Because your username is going to be different. You might have downloaded it in your downloads folder, most likely. So it might be somewhere else on your computer. And the way you've organized it also depends on your operating system. So this whole thing is not going to work. But how do I make sure that the same workshop material works for all 40 of you? That's why I have used this R project, which will first make sure that we are only using our relative paths going forward. And if all of you say here, what it is ending with will be where your R project started off. So every subsequent path that I give for either reading in or exporting files, it's all going to work like a charm. That's the advantage of using the here package, again by the awesome Jenny Bryan, and by using R project. It makes your work more reproducible and easy to share. All right.
So Lucas is asking, how do I add my or CID to the output HTML later? Do I need to install something more? Or C?
Okay, so that's an excellent question. So let's go to the, can we get to this at the end when I talk about rmarkdown? Because I'm adding this with rmarkdown. We are still doing the other packages now. Okay, all right. But the ORCID ID can be set here in the rmarkdown file. So that's your quick answer. But your long answer, I'll get back to it later on when we come to rmarkdown. Okay? Because there are a lot of things in this YAML part of my rmarkdown that I'm not talking about right away. All right. So we are done with the initial exploration of our data. We have the data set locally. So now we are going to part 2, which is using tidyr. So we are going to talk a lot about tidy data today. And Tidyverse is all about tidy data. So what do I mean by tidy data? I basically mean that every observation is saved in its own row and every variable is saved in its own column. Okay? You might think that every matrix, every data frame basically has columns and rows. So what is the big deal? What am I talking about? Right? So I'll give you some examples here. So for instance, in this case, country, year, cases, population, these are the variables, the columns, these are the rows. And when I say value, I'm talking about what is in each cell. Okay. And then we have the wide format. For instance, the data set that we currently have is in the wide format. I'll tell you again the difference between wide and long when we go back to our real data set. But wide format, basically, example, our case, has 57,000 rows or observations, and it has 25 columns. You might think I have 57,000 rows and she's calling it wide format rather than long. What does that mean? Right? I basically mean in the long format, I have a key value pair. So each column here is a kind of variable. I'm not spelling out each variable and throwing it into a separate column. Okay. And the way to go between the wide format and the long format is by using something called pivot longer. So if I have a really wide file, for example, I have 20 samples next to each other as columns, right? I have the AA colon, I have the AA lung, I have BA something else. And I have all of those in columns. But at the end of the day, what we know is that it's a sample. And then it has values. So all we need is which sample it has and which value it has. So we actually prefer a longer format, which is the tidyr format, which is easy for us to work with. So for that, to convert very easily from the wide to the long format, we will use the pivot longer. If you want to go back for some reason, you would use the opposite, which is pivot wider. You spread the rows to the columns, and we will see all of this in action. And the other couple of important functions in tidyr are basically separate and unite. So you basically have one column and you're separating it into multiple columns. And again, the opposite is you have multiple columns and you're squishing it into one column. Why would you do that? Might be your question. Right? When would I want to squish or separate? So the way to think about it is, so pivot longer is basically, for example, I have multiple samples here, right? AA colon, AA lung, BA stomach. Now these will become AA lung, AA colon, BA stomach, and all of that, and it will have the corresponding values. So this is what I mean by pivot longer. You want to go back, I just say, take the sample key value pair, put it back. It will get the column names from here, the values from here, it will populate it. So when would separate and unite be super useful? You can think of a very simple example. Now, let's say I have a column here, and just to keep it, compact, I might have a column that says date, right? But I might actually want to look at the sales on a monthly basis or a yearly basis. And here I might have 2021-04- today's date is 24. Okay. But I might want to separate that and say separate by hyphen and throw it into 3 different columns, which is year, month, and date. And then I can say, give me stats on a monthly basis or yearly basis. I could not have done that if I had them together in a date column. And if I want to do the opposite, which is at the end of it, I want a compact data set again, I might want to squish it back. So back to RStudio. Okay, so now what I'm going to go to is part 2. If you're not seeing this, you basically click on this outline here. Okay. So earlier on, until about 1 or 2 years ago, the pivot longer and the pivot wider were called gather and spread. But now the function names and the arguments have changed slightly in the recent Tidyverse. So now we will be using pivot longer and pivot wider.
Sorry for interrupting.
Sure.
Sometimes I had to switch from pivot longer to wider, or vice versa, when I was doing data visualization. So yeah, sometimes for data visualization, it's really helpful.
Exactly. So that's why when we do all of this and we go into the 3rd or 4th part of data visualization, we'll see how handy this comes. So that's an excellent point, Hedia. I see that Ali has his hand raised. Do you have a question?
Ali, do you have a question?
No, it is an old one.
Okay, sorry. All right. Okay. So again, why don't you follow with me for the first couple of minutes, and then you can try it on your own. So this is our data frame. Just to remind you again, if I just open my data frame, if it is in your environment, it's going to show up like this. Okay. The first thing that I'm doing is I'm selecting the columns that I'm interested in. Okay. So here there are many columns. But as I said, I want to take these columns and make the wide data set a long data set, right? Which means when I say I want this, it also means that I do not want this. So there are 2 ways to subset my columns now. So I'm saying, yes, I want the gene ID and I want anything that matches this pattern. And what is the pattern? Because it's within square brackets, it means or. I'm saying I'm either interested in column names that have A underscore or F underscore. So either this A underscore, all of these columns, or the columns that have F underscore. It's going to take gene ID and it's going to take this. Now let's just run that to see if that's what is true. You don't have to take my word for it. There you go. So now you've done a simple thing. Basically, you had 25 columns. Now you have 20 columns. And instead of specifying, you could have specified every column's name here, like gene ID, right? I could have said gene ID, A colon, A heart, A kidney. I could have done it 20 different times. Or I can do the simpler, smarter way of saying, hey, I know what the pattern is here. I'm interested in either A underscore or F underscore. And that's the easy and efficient way to subset your columns. Okay, now I've subsetted my columns. Now I want to pivot it to a longer format. So what I'm saying is take the columns that match this pattern and I convert it to a sample and counts. So now instead of having 20 columns, what do you think I'll have? Okay, we already have the answer. So we only have 3 columns because we have said don't do anything with the gene ID, only take these columns and switch them up. So now you have your samples and your counts, but instead you have it in a long format. Now you see what I'm talking about, right? I have 1,000,000 rows now instead of 50,000. This is why this is the long format and the other one is the wide format, not because of the 25. And this is a cleaner format or a tidier format because there are 2 kinds of data here. One the sample and one the counts. Now which sample is part of your data, right? And when you have it this way, you're able to extract that information very easily. But when it was spread across as column names, it was very hard to figure out what these were. So next what I'm going to do is, just because we can do it, I'm going to spread it right back. So I started with this, then I'm doing the opposite. So this is to undo whatever I did. I'm just saying take the output of, so until here, you will see the exact same piece of code that I copied from above. So each time when I show you things, I will only be adding 1 or 2 lines of code at a time. You don't have to think of this as, oh my God, there is so much code there. No. Each step, you're only adding 1 extra piece of information. So here, this already works. You get this. Now I'm adding 1 more piece and I'm running this now. When I run this, what I just get back whatever I originally had. And this is just for us to check that things are working properly. So I'll also show you unite and separate with this data set, and then you can go back and try things. So here again, I'm doing the exact same thing as before. Nothing new, nothing different. I'm just taking gene ID column because I always want to know the identity of which row it is, correct? So I'm always keeping my gene ID column. And I'm only taking the sample columns because the other one is a different kind of information. We'll come back to it later. But I'm just taking these counts columns. Again, you know what this means. I'm just changing it to a long format. But here I'm doing something different. So you see now there are 3 pieces of information here, right? It just says sample, but I have the first letter, which is the technology. The second letter, which is either it's adult or fetus. And then I have a third piece of information, which is the tissue type. So I have 3 pieces of information squished into 1 column. But I want to separate them and give them their own rightful column so that I can do something meaningful with it, especially when I'm summarizing my data, right? I might be interested in comparing how my colon tissues are doing as against my heart tissues. But again, in this format, it's very hard for me to do that. So how do I separate? It's actually pretty simple. I take the sample column. So that's what I'm saying here. Take the sample column. And then I'm saying split the sample column or separate the sample column into 2 different columns, which is the source and the stage. Again, I'm putting them together. And here, the other second column is my tissue column. And I'm saying in this case, my separator to be considered is underscore. Okay. And now let's run it and see if we are getting whatever we are expecting to get. And my RStudio is slow. Really slow. Okay, while my RStudio waits, I'll tell you one more thing. So you may have noticed that I'm using something like this. Yes, we can, Sumit, but I'm not using it here because actually it's not that big a data set. I just have way too many things open. That's why it's failing. But yes, we can do it. All right. So first, let's look at our output here. We wanted to separate it into 2 columns. Yes, we have 2 columns. We wanted to separate by underscore. So AA underscore colon has been split. All good so far? These are the two new column names. The corresponding counts are there. So we have exactly what we want. Okay, now let me go back to what I was saying here. So this is called a pipe function. And I'll tell you what a pipe function is. Now, if you're familiar with, say, shell scripting or Unix programming, you might have heard of pipe. So I can do something simple like this. I might say, list all the files in the directory and then do a head of that, right? And this is called a pipe. And what pipe operator basically does is it maps the output of one function to be the input for the following function. So I can keep piping things as much as I want here. So I can say, take a bunch of files, or you can grep something, right? Meaning I'm looking for some patterns, and then I can pipe it to something else. Or I can save it to something else. So basically the pipe function lets me add multiple functions without having a complicated nested looking thing. Right now, if you want to look at an example for R, right, I showed you an example somewhere here in the previous part. For example here, right, I said view head this, and this is basically a nested function, correct? So what I'm basically doing here is I'm taking this function, rather, I'm taking this data frame, I misspoke. I'm passing it to head, and I'm saying, take the first 100 rows instead of the default 6, and then I want to view it. But if you think about it intuitively, how does the human mind work? We are very used to reading things from left to right or from top to bottom. We are not used to reading inside to outside. That's not the way we typically read. So what the pipe function does is basically to do the exact same thing, but in a much cleaner way, right? So what we can, so for instance here, let's take this, right? What I could have done is something like this. I could have said, select my data frame and then this, and it would have worked. Now on that, if I want to run pivot longer, I could have passed, so here, right, I could have passed this entire thing and put it inside pivot longer, but very soon you will realize that it becomes unreadable. How do I keep my script clean? So you can think about this pipe function. You can read this pipe function. So this is the R pipe function, which is part of the Magritte package, which is also part of Tidyverse. So the main thing that this one has is this pipe function. Okay. And the advantage of having this pipe function is you can read it simply in English as take the data frame, then select the columns, then pivot it longer. Or in this case, I'll say, take the data frame, select these columns, then pivot it longer, then go back to the wide format. So you can read it as then. So it makes it so much easier to understand your code, and more importantly, understand someone else's code. If I had given you this nested function, I'm pretty sure you would not have come back to any of my subsequent workshops, right? But now we have this beautiful pipe function that makes our code so much more readable. And I can add these nice little comments at the end to say what each step is doing. So I would always encourage for you to use these pipe functions. Now, naturally, they work very well with Tidyverse functions because it was written that way, but it works with most base R functions as well. The thing to remember is, as with your any other pipe in any other programming language, it will pass your output to the first argument. So if you want it to be something else, you might want to be careful about that. Okay. So by default, the first argument for all of these are the data frame or the data. So in this case, it's working out well so far. But in case your pipe isn't working, that's something you may want to look into. All right. So we separated. All good. Oh, this is the one that was taking long. Sorry for running it again. Oh, thank God. Okay. So what we are doing next is, I have my source and my stage. Still, it's part of the same column, but I want to separate it further. So the cool thing with separators, there are many different ways in which you can separate. You can either say separate with an underscore, or I can say separate by character position. Now we know that it only has 2 columns, right? This particular column, source underscore stage, has only 2 characters. So I'm just saying the separator is after 1 character, which basically means I'm expecting it to split after the first character. Now let's see if that's what happens. And let's hope it happens fast. So because this is taking a while to run, this might be a good time for you to get back to the tidy portion of your rmarkdown as well. If you're already following things along, that's great. If not, I would recommend that you try these things. Because there is only one more function that I want to show you here for this part, which is unite, which is similar to what we did with pivot longer and pivot wider. What unite does is I'm telling it, take these 3 different things, put it back together. Or take these 2 things and put it back together. Now, if and when it runs, oh, there you go. See, I just said separate after the first character and it does exactly that. So I have my source and my stage and my tissue. And as I said, I'm only adding 1 more row here, extra. Everything else, I'm just copy pasting from the above one. Okay. Now see what we are doing here. We started off with 3 pieces of information. We split it in 2 steps. And then finally, when I'm using this unite, I'm trying something different for a change. I'm saying, okay, the source is something else, it's a technology. I'm not bothered about that. Let it be separate. But I want to combine the adult or the fetal thing along with the tissue. So I'm combining it this way. So now if I unite, instead of having 3, I'll have 2 columns, and the second column will actually be a combination of my stage, which is adult or fetus, and the tissue. I think somebody has a question. No. Oluwafmi, do you have a question? I'm sorry if I'm not pronouncing your name correctly, but do you have a question?
Yes, thank you, Ma. I have a question, Ma.
Yes.
Okay, my question goes this way. Based on the pipe you just explained just now, is it possible that I use a pipe to pass a function to a linear model? Will it work?
Yes.
Okay.
So we are not using tidy models today, but it can be passed to most R functions. Yes, you can use it.
Okay.
As long as you keep in mind that the output of one is compatible with being the input of the second function that you're using, it should work fine.
Okay, thank you, Ma.
No problem. On, please, you can just call me Janani, by the way. Okay, so here we have combined the stage and the tissue, and we have this new column the way we want it.
You know, she was busy with her screen. She never even checked. Not your girl tell and inform.
Sorry, the mic. Can you mute your mic? Thank you.
Okay. So with this, part 2 is done. So I will give you a couple of minutes to try. If you have any questions, if you're stuck, or if you want to talk about something else related to these tidy functions, this is a good time. Yes, Jana.
Okay, so thank you. This has been a really wonderful session so far. I just want to make one comment. It was interesting to learn about the separate function, because before I think I was trying to think about strings and separate using the stringy package. And this is much cleaner the way you've presented it, I think.
So the stringer package is actually really helpful, but
yeah,
as you said, it works great when you have limited set of strings. But in this case, you're working with entire columns, right? It's much more efficient to do it this way. That's the main thing.
Yeah, thank you.
Am I pronouncing your name right? Is it Jonel or Yonel?
Jonel.
Jonel?
Yes,
okay, cool.
That's right.
All right.
Thank you.
Mona? Yes? Oh, sorry. That was the raised hand of Jonel, not Mona. Everyone else doing good? Is the select working as expected? Pivot longer, everything else looks okay?
Everyone is saying yes.
Okay, cool. We can move on then. Part 3 is another very powerful package with few more functions, which is dplyr. This was built upon another package that was plyr, but now all of this is again part of Tidyverse and Tidy Data. So the good part is you've already used select. You already know one of these. So the first two functions are to pick rows or pick columns. So when I say rows, basically filter is to select, or I shouldn't say filter is to select, it's confusing. Filter is to subset observations, or in this case, rows. Select is to subset variables by their names or positions. And these are basically the functions that will work on columns. And you've already seen a couple of examples of how to use select. So they're extremely powerful. Together, you can subset your data any which way by just using this combination of filter and select. And because they allow pattern matching and combining different ways to subset, they're really a lot of fun to use. The next two are mutate and transmit. So in mutate, basically you can create new variables, which are functions of existing variables. For example, if I have GDP per capita, meaning per person, and then I want to convert it to gross GDP, and I have another column which is the total population, I can mutate and create a new variable which is a product of these two, right? And transmute is a slight modification of mutate. So what mutate does is it creates new variables based on these other existing variables, but it keeps the old ones too. What transmute does is it calculates these new ones and it drops all the original columns. And I'll tell you the advantage and disadvantage of using these two when I go through the examples. So Baser has a function called sort, as does most of the programming language. So the Tidyverse version of it is arrange. It basically helps you reorder your rows or sort your rows by one or more columns or values in the columns. Okay. Group by and summarize are often used together. And I'll show you why this is extremely important. For example, let's say for instance, I was mentioning, right, that I might be interested in my monthly sales or annual sales. But if I'm getting my average across my entire data set, it's going to span years, months, it's going to span different geographical locations. But I might say, hey, that's not what I'm interested in. That mean doesn't even make sense. What I might be interested in instead is what happened in 2007 versus 2017. Was there a bump in this 10-year margin? Or what is happening in Oceania versus Africa? So then what you will do is you'll group your data, which is you'll group different subset of your rows based on a particular variable. And then you'll say for those groups of variables, now you summarize the data for me. Either give me the total, give me the mean or median or whatever that is. So when you use group by and summarize, you can very easily and dynamically generate summaries for different subsets of your data set. And again, we'll see all of these in action. So don't worry about if something is unclear now, that's okay. It will resolve itself. So these are the pictorial representation of how filter works. I'm just subsetting rows, I'm subsetting columns. If I just used summarize on the entire data set, this is how it would look like. For instance, this might be the total, the mean, and the median, right? But here, when I say group by, and let's say the blue ones are Oceania and this one is Africa, then I'm going to have summary for Oceania and I'm going to have summary for Africa. And this one might be for Asia, right? So basically, I use group by and summarize together. There are some other functions that you might be familiar with, which is when you have multiple data frames, how do you work with them, right? You have two different data frames here, for instance. So you have joining functions. So you have left join, where you're keeping this one intact and you're tagging this along, or you have right join, you have inner join, which just takes the ones that are common to both. You have outer join. We will not be doing demos of this, simply in interest of time, but feel free to explore this. And these are some really cool tidy explain GIFs created by Gedenbui, or rather Garrick Adenbui. So this is how left join works. This is how right join works, keeping the right one intact and dropping anything that's not here. This is how inner join works. It only keeps the ones that are common to both. And outer join basically takes them out. And then you have your traditional set operations that you can again definitely do within Tidyverse. So the most common ones are intersect, union, and what's the difference between the two data frames that you're comparing. So again, let me show you the GIF versions of it because it's quicker. So intersect, it only keeps the ones that are common in the two intersecting sets. You have the union, which is it takes everything and puts them together. And then you can bind. Again, there are base R versions of bind rows and bind columns. But whenever there is a Tidyverse version of doing something and there is a base R version of doing something, I would personally always recommend the Tidyverse version of it. One, because it's cleaner, it keeps your data frame format, your tibble format consistent. It gives you meaningful errors. For example, if you're combining rows and the column names are not even consistent and you didn't notice, it will throw an error for you. So it's generally helpful. I will very strongly recommend using Tidyverse over Baser whenever you can. So standard, very good examples are using bind underscore rows and bind underscore columns. And now we go back to RStudio. I'm starting with part 3, which is if I didn't move things around. Okay. Oh, never mind. Part 3 should be 261 row or line, sorry. All right. So just as a reminder, this is the head of your data, because you may have forgotten that these other columns also exist. Now I'm bringing back these other columns. I told you I'll come back to it, right? So now we are going to use the other columns when we are experimenting with these new functions. I said this is the length of the transcript, right? This particular column. So what I'm doing here in 280 is I'm basically taking the data frame and I'm filtering and I'm saying I'm only interested in these really short transcripts. So for instance, here you see there are many that are 2000, 1000, 1700. When I filter, I only have the tiny ones. And then I say this can be rewritten with pipe because, well, that's easier for me to read. It's the exact same thing. I say take the data frame, then filter it in such a way that the length is less than or equal to 50. And it does exactly the same thing. See, I have the same 124 by 25 in both cases. All right. So this was a numerical way to do it. Now let's say I want to, so there are multiple chromosomes here, right? And for those of you who are familiar with biology or maybe even otherwise, you know that there are 23 chromosomes. You might ask, why do I have, okay, that's the different thing.
Sumit is asking a question, sorry.
Oh,
so
yes.
there be any difference in GS, sorry.
Nope.
Adding the length and dollar length.
No, so if you're using base R, base R, you would be using the dollar to tell which column you're working with. I should have mentioned this upfront. Sorry, it's my fault. The really cool thing about using the pipe function and using Tidyverse is once you've read the data frame in a table format, it already knows what your columns are. So you don't have to say GSC dollar this column name. It knows when you say length. In fact, this will autocomplete. Can you see this pink token here instead of the blue one? These blue ones are all functions. This pink token is telling me this is a column, and it automatically shows me all the columns in the loaded data frame. So I don't have to use dollar length. That's the other beautiful thing about using Tidyverse as against using base R.
So generally, in some of the formulae I have seen, primarily with Ggplot and all those, sometimes the tab does not work, and there what I generally do is I use the dollar formula so that it gives me the tab.
Yeah. So if you're subsetting it differently, or sometimes you have to factorize, and it's not very clean to know which columns you're working with, then you might want to explicitly mention using dollar. But in all of these, I'm doing straightforward things. I'm just saying take it and filter by a column. So in these cases, it is superfluous. I don't have to use the dollar. But as Sumit mentioned, if there are more complicated usages, then and it's not working, the autocomplete is not working, and you're not sure, you get an error, then specifically use the data frame dollar, the variable name or the column name. That will resolve your error. Okay, but for the time being, you will notice that I'm not using the data frame each time. I'm only using the column names directly. And the other cool thing is, in base R, I would have to throw all of these within quotes. Tidyverse understands that these are column names. It treats them as variables. It does not need quotes. All right. So the next example of filter is I'm saying there are many chromosomes. There are 22 somatic chromosomes. And then there are the X and Y chromosomes. And then I think, in fact, this data set also has the mitochondrial DNA. So there will also be chromosome M, which is the mitochondrial stuff. Anyway, I'm only interested in chromosome Y. So can I subset by that? So again, here you will see, because it's been sorted a certain way, you have chromosome 1, then 2. But I'm saying I only want this. And grep L, if you're new to all programming languages, then what grep L does is it basically does pattern matching and subsetting. If you're used to other programming languages, this is the R version of it, and I can call it within any other function as I'm doing here. So the arguments are, I'm saying what the pattern is. So I'm hiding the arguments, but you can, if you're still new to the function, You can say pattern equals R and X, meaning this is the pattern and this is where I'm searching for it. That's what it means. Okay. Now I run this and I only have 540 rows and all of them are from chromosome Y. Of course, the length goes back to the other ones because now I'm not using the length. But I can also do that if I want. And there is just one row, meaning there is only one short transcript from chromosome Y. Now, why am I interested in this transcript? Well, right now I'm just demonstrating it. I'm not particularly interested in this transcript. But let's say I am interested in small RNA regulation, then I might be interested in doing this. If I'm interested in ones that are only on chromosome Y versus X, and if I'm interested in small RNA for any detection or diagnostic purpose, then I might be using a very simple filter function like this. Okay. Now select you're already familiar with. Remember I had said you can use square bracket to indicate OR. In lines 301 and 302 is I'm taking my data frame. I'm always selecting my gene ID because that is my important identifier. And then I'm saying select columns the .f underscore, which means I'm only interested in my fetal samples. I'm not interested in my other samples. What does dot mean? Anyone who's used grep or regex from any other language wants to tell me what dot means?
A wildcard?
Yeah, it basically means it can be anything. In this case, it can only take 3 values, A or O or I think B. It can only take 3. But just to be safe, I'm saying it can be anything. And dot also means one wildcard. It means one character that can be anything. Okay. So now when I run this, these are the only samples. There are only 4 that are fetal samples. Okay. And this is another way to use select. So far in these examples above, we are selecting what are the columns we want. But another way to select is to say what are the columns you don't want. Sometimes what you want might be a cleaner list. Sometimes what you don't want might be a shorter and cleaner list. So you can use any combination of these. But in this case, instead of, let's say I'm not familiar with regex, or for some other reason, I say I just don't want these columns. So I'm spelling them out. I'm saying I don't want the chromosome start, end, length, anything to do with the gene metadata. I'm only interested in the gene ID and the sample data. I'm not interested in this other metadata. So please remove that for me. And when you do that, it removes those first 5 columns after the gene ID. So this is another way to select. Now let's say you want to exclude with the pattern. I'm doing the exact same thing as before, but I'm adding a minus that says exclude. So now it only has the details for the gene and the chromosomal location. It does not have the other sample data that we were looking at. So it is as simple as toggling this minus. So if you want to select a few columns and then filter, so here what am I doing? I'm saying chromosome Y, right? But why am I saying chromosome 1? That is because I'm using OR. This is to say OR. So if I'm looking for multiple pattern searches within grepl, the pipe is for, this vertical pipe is for OR and the ampersand is for AND. So if I want both conditions to match, I will use AND. Otherwise, I'll use OR. So now the reason this has been picked up is because it's less than 100. There might be others that are being picked up because it's chromosome Y. But this is the strange way that I have decided to subset today. Maybe I can try this. And now it's giving me a combination of these two. So I'm saying it's chromosome Y and the length is less than 100, less than or equal to 100. Okay, we can take a minute. Are you all trying this with me? If you're not, why don't you go ahead and try, ask me questions, and then we can go on to mutate and transmit.
I will ask a question about regular expression. So sometimes I need something in the middle, it's like sometimes you have a gene ID and then you need the specific like pattern. So how, for example, to tell Tidyverse and R, I need only this in the middle or after this or before this, pattern?
Okay, so we will do regex a little more in the next section. So maybe that will help. You'll see a lot of regex coming up in the next few things. But to give you a short answer, some simple things to remember about regex in R is basically dot, as I mentioned, is for any single character. Any single character, okay? Now star is for any single character, but any number of such characters. Okay, so now let's say I have this. Now what it's going to find is anything can precede pattern and it's going to pick this up. Okay, but you're saying it is in the middle, which means anything can also come after this.
Okay.
So if I didn't have the star, it will probably look for patterns. It will only allow for one more character to be there. But if I'm saying anything and any number of characters can come after pattern and you don't know what that is, this gives you the flexibility. So this combination is very powerful. It says any character and any number of times.
Thank you.
So this will give you the exact subset of picking out your pattern in the middle. But we will look at some examples and hopefully it helps.
Yeah. Thank you very much.
Oh, no problem. All right. Before we go to mutate, last call. Any questions on what did we do? Select and filter. So either this is too fast or too slow or too boring or everything is amazing and perfect. I choose to pick that you're all very happy with me. Okay, I'm moving on to mutate. All right. So what I'm doing here in the mutate part is I said you can use mutate to create new columns. So for this, again, I'm doing the first part. I'm just checking. See whatever is selected, I'm running it with command enter. Since you can't see what I'm doing, my shortcut is either command enter or control enter. I don't want to run the entire thing, so I'm just selecting this without the pipe. The reason I'm not selecting pipe is if I select pipe, then it will expect for me to say something after that. I don't want anything else, so I'm just running this. Okay. With that preview, I'm just saying everything minus the sample data, so I only have this. And then I'm running this, which basically, instead of giving me 57,000 rows, it just gives me the top 10 rows. And then if I run the entire thing, it opens it up in view. So I'm just running it step by step to show you what is changing. Okay. Now this is what Hedia asked. But don't worry about these different regular expressions. Don't worry about so many lines being there. I'll take you through line by line. So the first thing here that I'm doing is the same as before. I'm just selecting my sample data. But the difference is, see, it's not printing anything to the screen. That's because now I've become a little more intelligent. I said I'm doing all this work, but I'm not saving anything. So I'm having to repeat it again and again and again, right? I'm copy pasting my code so many times. It's not effective. It's not efficient. It doesn't even look good. So I'm saying I want to save it now. So this is the way to do it. You basically use your assignment operator and you throw it into a new variable. So now if I look at gene, it's the same as before. It's the same as before because, well, I only ran these 2 lines, right? Anyway, so I run that. What am I doing next? So here I'm saying I'm selecting columns. Next, let's first see. I'm doing something with gene ID, right? And I always said, look at your data first. Because when I'm doing this regex, something is going to change. But you don't even know what this looks like because of the ... So first, let's look at what it seems like before we change anything. Okay, so this is how my gene IDs actually look. For the first time, we are getting a clear picture of it. So it is alphanumeric, no special characters, good so far. It's starting with something like this. It has a string of zeros. It's basically the ID. And then this dot something at the end. For those of you who know biology, it means these are isoforms. So those of you who don't know biology, don't worry about it. It's just different ways in which your genes are transcribed. The details are not important for this workshop. Okay, so that's what I mean by remove isoform number. So what do I want to do? I only want to keep the gene ID and I don't want to keep the isoform identity. Just to simplify things, right? Because I might have multiple isoforms, but I probably only want to know which gene. At this point, I don't particularly care about which isoform. So I want to remove this dot something. So it takes us back to Hedia's question of how do I remove it? Because what is coming here is changing, right? So there is not one particular pattern I can specify and say remove that. And I can't say remove the last 2 characters because sometimes it's the last 3 characters. So what would you then say? What is common to all of this? The one that is common to all of this is there is a dot and there are some number of digits after that. Okay, so then this is how I'm saying. So G sub is to subset or it's a some substring search, sorry. So I said in any pattern search, dot means it can be any character, correct? But in this case, I want the actual dot. I want the actual full stop or the period. So these double slashes, in other programming languages, it might be a single slash, but for R, it's 2. This is my way of saying, I don't mean any character, not that kind of dot. I'm interested in the actual dot itself. So I'm basically escaping the character. Yeah, that's the escape character, correct. So it's a double slash. And now when I select for this, what it does is it goes to everything in gene ID and it finds this escape character. And then I'm saying there has to be a digit after that. And it can be any digit, 0 to 9. So again, it's within square bracket to say any one of these things, doesn't matter which. Finally, I want it to pick up 13 as well as it picks up 1. So I add the star. So I'm saying any digit and my star says any number of digits. Okay, so now see what changes. Okay, I'm not going to save it to the variable. I'm just going to run it on the screen. Oh goodness, you can't see it, can you? So I'm just going to select this gene ID column alone so that you can read what's there. You see, it's cleaned up. It doesn't have the isoform information anymore. So that is what you've done with this first regex here in line 332. Next, it seems like I'm not happy with my chromosome column. Let's see what's up with the chromosome column. This is why I'm not happy with it. See, for each transcript ID, there are multiple start and stop locations here, right? These are all part of the same these are all part of the same isoform of the same gene, therefore the same chromosome, but there are multiple start and stop sites. So multiple pieces of the transcripts are being read and they're all being squished into one row. Now this is really confusing. Now, for instance, I can't even subtract and calculate the length, the end minus start. I can't calculate that because which end and which start do I subtract, right? So I want to do a very simple thing here. I'm just saying I'm only interested in the first entry. From every column, I'm only interested in this first one. So it will take the chromosome 1, it will take the corresponding first start, it will take the corresponding end. See, there are many, and this will all actually be the same number. But I'm saying I only want the first. So that is what I'm specifying. And here our delimiter, as you can see, is a semicolon. And you see, chromosome, start, end, strand, all of these basically have these multiple entries. Now, length only has a single one because it's not depending on the start minus end. It's depending on the gene ID. All right, let's get back here. So what I'm saying here is clean it up. Take the chromosome column. Why did I use this? This might be what Sumit mentioned. I'm sure I can run it without that too. Oh, no. Because, no, I can run it without that too. Okay, so what I'm saying is, take the semicolon and remove everything after that. So that is how I want to specify my regex. I'm saying, go to the first semicolon that you see, remove everything after that. I don't want to see it anymore. So what I'm doing here is I'm basically taking the semicolon, any character, any number of characters up until the end. Dollar means end of the string. So caret means beginning of the string. Not that beginning. Okay. Not that either. Of the string, and dollar means end of the string. Okay. So when I use it in regex at the end, it means go all the way until the end of the string. And this next, I didn't mention this, sorry. This substitute, what I'm doing is for the substring match, I'm basically taking this pattern and I'm replacing it with nothing. So you see there is not even a space there in quotes. So I'm just saying take it, replace it with nothing, which is as good as saying remove it. So here what I'm saying is take the semicolon and everything that goes after that until the end of the string, replace it with nothing. And let's see what that looks like. See, now you don't have the semicolon and all that after the chromosome. It only keeps the first one. And then I do it again for the next column, which is exactly the same thing. But because it's a numerical column, just to be sure, I'm using the as.numeric. Because this, it's just treating it as a character string. So this is just to be safe. That's the only difference here. I'm using the as numeric because my start and end. So I want it to be double. So I'm converting it. See, when all of these are stringed together with the special characters, it is considering it to be a character variable. But I want it at the end of the day to be numerical. So I'm saying cut it all off and finally convert it to numerical. So it says double. Why am I doing it? It's for my future operations because I might want to subtract one from the other. If it's character, it's not going to do anything. If it's numerical, it'll actually do the subtraction for me. So I'm just making sure I'm thinking ahead. Okay. And then I'm doing this. I'm doing the same thing for end. and I'm doing the same thing for strand. So now when you see it next time, you should have only one entry for chromosome, one for start in double, one for end also in double, and in strand I should only see the first one. Let's see if we are getting exactly that. There you go. You did all of that in one shot. And you've cleaned it up. The first step of cleaning up at least. And I want to save it to a variable because I'm going to call it later. And I'm just saying, show me the first 10 rows. This is the entire thing. This is, I'm saying, show me the first 10 rows. Okay. I'll show you a couple more examples of mutate. So for instance here, I'm just trying to make it a little more readable. So these are all in basis. I want to make it kilo basis, so I'm just dividing it by 1000. So far, if you go above, you'll see that I use the same name as the input column. So I've actually not created a new column in some sense. I've basically replaced the column with the cleaned up version. But in this example here, I could have used the same start, but I want to keep the old one too, and I want to keep the new one, which is the kilo base version. So in this case, all I'm doing is I'm taking the column, dividing everything by 1000, and I have these new columns now. And as I said, mutate will keep the new ones as well as the old ones. But if I didn't want the old ones anymore, I would just use transmute and it will automatically drop the old ones. Now this is not exactly helpful because it has even dropped the gene ID and I want the gene ID. So I might ask you to add that back. Let me go over a few more functions and then you can, it's already past 1.5 hours. After this might be a good time for break before data visualization. Okay, so just give me 5 to 10 more minutes. So distinct is similar to unique in BaseR or in other programming languages. So you just want to know what are the unique values that chromosome can take. It basically goes from 1 to 10. You want to let's see if this works. Nope, it doesn't work. Oh, maybe that's coming up next. Okay, so here I'm just getting the, so these are the 25 different values that it can take. As I said, the 22 chromosomes, X, Y, and M. We'll look at that in a bit. Same way I can do it for strand, but this only takes 2 values, plus and minus. And then I can get unique combinations. So this is basically 25 into 2, pretty much. So this is what I was trying to do. So you can sort this in descending order. So far you only saw chromosome 1, right? But now I'm saying reverse sorted by chromosome. So it's starting with Y. And within chromosome Y, so here it's basically reverse sorting by chromosome. And here I can reverse sort it by chromosome or just sort it by chromosome and length. Now you see that it's chromosome 1 and it's going up in length. So I can again, the way I did it for distinct, for any of these actually, I can do it for combinations of variables as well. Okay, let's take a couple of minutes. Why don't you try these out? We'll come back to group by and summarize. Because I've been talking for a long time, might be good to hear your voices.
Janani, I guess the descending order would not work on characters, right? Or would it?
Sorry, I couldn't hear you very well.
Yeah, the descending order arrangement on descending order, it would not work on characters now, would it? Row,
That's right.
line 373. Yep, 373.
It does work on characters. Actually, it's only the minus or plus that might sometimes not work on characters. Now, instead of descending, if I said minus, actually that might also work. Tidyverse always surprises me. See, it doesn't like minus because that's only for numerical values. But because it's character value, I use descending and then that works. But I can use the minus for any numerical column or a double or integer kind of column.
Thank you.
Janani, so how about look around, like look ahead and look behind? Regular expressions.
Could you tell me what you mean by that?
Actually, I use it a little bit. I don't know how to. I will put the regular expression in the chat.
Okay.
And you can tell me if you are used to it.
Okay. No, actually, I have not used that in R. I've only used that in Unix. So if you want to go ahead and tell people about this in R, please go ahead.
Actually, it's the same as you said, but instead of putting the dot star, it's using these two regular expression. So the first one, it's look ahead. So it looks ahead, you know, it's like a certain pattern. And then, yeah, look behind. So it's like if you put it and you can look, yeah, like for a certain pattern. And it's quite helpful in genomics.
I see. Okay, this is really cool tip. Thank you.
Yeah, thank you very much.
Okay. So everyone with me so far, please raise your hands just so I know you're still here. Okay. And things are working for you as well, not just on my screen. So you believe that these things can work? All good? Okay. Can I proceed to group by and summarize? All right. I'll take Stephanie's word for it and move on.
Yeah, they are saying yes in the chat.
Okay, very cool. Okay, thank you, thank you. All right, let's go on. Also in interest of time, because after this, you can go to your much awaited break. So group by and summarize. So as I said, I might be interested in getting summary statistics, but for subsets of my data, not the entire one. In this case, I might want to know what are the longest or the shortest genes, or what's the gene length distribution across different chromosomes, which means I first have to group by chromosome. So this is my entire data set, right? Now what happens if I say group by? No summarize, just group by. It basically says there are first and foremost, it's kind of sorting them, and it's telling me there are 25 groups based on the chromosome column that I've picked. Okay. And what I'm doing next is the summarize function. There is a British and American version of it with either Z or S. Use whatever you like. And if you use the summarize function, basically I can say this, what this N does without any argument is it basically counts the number of entries. Okay. So if I just run that, it calculates a new column called number of genes for each group. It basically counts the number of entries. So it says there are 5,000 gene IDs in chromosome 1, 2,000 here. And see, this is a strange way of ordering, right? It says chromosome 1, then 10. That's because it's not considering these to be numerical. These are all characters. So it says 1 and nothing, 1, 0, 1, 1. And then after it goes through 19, then it starts with chromosome 2 and 20. So it's very strange that way. Anyway, but there are multiple summaries I can calculate in one shot. Oh, sorry. I forgot that I added this extra bracket. So now I'm doing two things. I'm calculating the number of genes and I'm seeing where is the first gene starting. Right? So there are different start locations, and this is along the chromosome. And when I say minimum of that, what does it tell me? Right? So there's a gene, there are different start locations. What would the minimum mean? It basically means the first gene or the first transcript on that particular chromosome. That's why I'm calling it start of first gene when I calculate the minimum of the start column. Okay. Here again, what I'm doing is I'm same grouping. I'm just adding two summary variables. Now I can do other things too. So now I'm sorting by length here. And I can group by two different variables. And as I said, this group would create how many? 25 into 2. So there are 50 groups here. Because it's creating groups for this combination of chromosome and strand. And now that I have the correct combination on both the strands, so what I'm saying is I'm interested in everything to know. I'm interested in the smallest gene, which is basically I'm sorting it by length, right? So if I take the first gene ID, what will it give me? Because it is sorted by length, if I didn't have this, this would give me the wrong answer, right? So because I'm sorting it by length and I say give me the first, it gives me the smallest gene. If I reverse sorted it, then that would give me the longest gene. I'm getting the maximum length, which is the longest gene here again. But the actual value of the length of the gene. And then these are basically my interquartile ranges. Let's see what this looks like. So you get this for each combination. Okay, number of genes, smallest gene, and the longest gene you can't see because I've restricted my screen width. So all that I did here, I'm not saving, but if I want, I can save this too. But for the time being, just the gene location, right? This is gene lock. You see this has 25 variables and this only has the gene location information. So what I'm doing is I can write it. And I'm saying go to the genomics for here, which is basically, again, to remind you, here basically takes you to wherever your R project is or where you started off. And then it goes into the genomics. And it's going to write this file there. I already have the file, so I'm not going to run it, but feel free to go run it. You will have it too. And then there are more things that you can do here, which is you can select, again, the other columns here, right? And there are more regex examples. If you want to create the counts into counts per million, how would you do that? You want to create a log CPM. So all of these conversions and examples I've shown you here. And that would be under the more wrangling section. And in the interest of time, I'm going to let you explore that. And we'll directly take a short break and come back for data visualization. Those of you who want to go take a break, please take a break. We'll regroup in 12. Okay, 12 is what, 6 minutes.
Yeah, at 5 p.m. here, Tunisian time.
And it's 4 p.m. GMT.
Okay.
Yeah. Whichever time zone you're in, and we'll be back in 5 to 6 minutes. Okay. And Sumit has a question. If any of you have questions, feel free to stay here. Otherwise, go take a break. I've seen a lot of places, a lot of grouping, people use ungroup right after. May I know why it is right after or not? What happens if ungroup isn't? Okay, I'm not reading out the question because all of you can read the chat, okay? What happens if ungroup isn't done after grouping? What happens? Basically, they remain groups. And for your subsequent operations, if you don't want to consider those groups and you've forgotten that you've grouped them, then that's going to mess up your summarize function. So that's the advantage of ungrouping. In these cases, so far, whatever I showed you, I just did an ungrouping or grouping, and I'm saving them as different variables, or I'm not saving them at all. So for me, there is no downside. But if you are saving them to a variable, then it means it will remember those groups. If you wanted to remember the groups for the different summarization and visualization, you keep it. Otherwise, you ungroup it. At any given time, if you have a question, you have a doubt, you can just go ask what the groups are. So that shouldn't be a problem. You can always reverse it at any point or regroup it at any point.
Yes, for me, when every time I use group by, I try to use ungroup. So then, I don't have a problem.
Rest of you, everything making sense so far? Any questions? Yes. I see one raised hand. Please ask your question. Oluwafemi. I'm sorry if I'm not pronouncing it right, but please ask your question. Okay.
I think everything is fine.
Okay.
No question. Everything's fine.
Yeah, that's good.
It's really interesting.
Yes, Janelle.
Thank you. So maybe I just have one genetics question.
Okay.
Let's say in this case we have several isoforms, and I think we chose the one that started with the lowest start number, right? So I'm just wondering, how do we know which isoform to keep generally? Because what if one is functionally,
Yeah, so normally the best way to do this might be to keep all the data. What I'm doing is not the recommended way. This is just to demonstrate that I can do a lot of cool things with Tidyverse. I'm just throwing out data left, right, and center. That's not the way to do it. So, okay, I should add that caveat once people come back to. It's not good practice to throw out any data that you don't like or anything that doesn't look clean. But now, for the sake of simplicity, I'm just restricting to the first one.
Great, thank you. Thank
you.
Yes, thank you.
Okay. And Okan, to your question, install.packages, Tidyverse. So if you go all the way to the top of my rmarkdown, which is line 88 to 90 is where you're installing these packages, right? But I add something else here, trouble installing Tidyverse, line 96. So it might depend on your R and RStudio version. Sometimes Tidyverse may not be compatible with your version, especially if you're using an older one. In those cases, I would recommend that you install the individual packages that are within Tidyverse separately, one by one. So you can, so far we have used Reader, we have used Tidyr, Dployer, Regex. Actually, we have not really used Regex. And we will be using Ggplot for data visualization in part 4. So if Tidyverse installation fails, you can install these 4 packages individually and see if that's giving you an error or not. Or you can update your R version, and that might also help.
I think he's using the R version 4.
Oh, you are using R version 4. Oh, correct.
Yeah, but I think he has to select the CRAN mirror. Maybe that's the problem. Did you select the CRAN?
So it'll give you 4 options, either cloud or something else. All, I think you have to select. Good point, Hedia, correct. Make sure you're selecting the correct CRAN mirror.
No, it happened to me so many times. So
shall we try installing it? Oh, people might be back. Okay, let's just take a minute to install. Oh no, don't, please.
maybe it's better to
not do it.
Not do it.
But there's a way to do it. Okay, so you can say force install and then dependencies equals true. You can use all of those options, even if you have something installed.
Maybe you can stay after, and then we can help you with the installation at the end of the workshop.
And if for any of you Tidyverse is not working, I would recommend you can follow along by looking at, where is the HTML? I think this. Okay, one of these HTMLs. Okay, this one. You can open it in your web browser. It will look like this. So I would have run it. So whatever I'm seeing on my RStudio screen, you should see the outputs here too. You should see the exact same outputs there, in fact. So you can use this as your proxy and then later resolve your issues and then go look at it. Okay. So whatever you're seeing on the console, you should see here, because I've pre-run the outputs and I've loaded them here. Okay. So it looks like, I mean, this is great. Having questions is great. And that's how you understand what's happening. But it means that we might go over 3 hours. So I might be rushing in the data visualization part. But if you have the time to stay beyond 3 hours, I'm happy to be here. And I can take more questions. Okay. Yes, Janal, one final question and then we'll move on. Yes, you have a question?
Excuse me, my mouse just went over the button there. I have no question.
Okay. No problem.
Sorry.
Excellent. Sorry. No problem. That's good. Okay, so we'll go to the last and final part, which is the fun part, which is, okay, you've done a lot of summarization, grouping, you've cleaned up your data at least a little bit, and you want to see your data visually, because that always gives you the best idea of what your data looks like. Okay, and we are going to use the Ggplot package that comes with Tidyverse to do all of this. The Gg in Ggplot comes from geometry of graphics. And there are 3 main components when you define each Ggplot function. First, you must have, of course, your data or your data frame. Next, you specify the aesthetics, or well, which columns are you plotting? Is it a 2-dimensional or a 3-dimensional plot? Right? And then the third one is what kind of plot? What is the geometry that you're using? Is it a scatter plot? Is it a trend? Is it a box plot? or is it something else that you're plotting? Okay. And then you can, now that you're using group by, right? So you can say, you can color something by one particular variable, or the shape can depend based on the other variable, right? The shape can probably be plus for plus strand and minus for minus strand, or the size can be proportional to the length, right? So you can add a lot of these aesthetics in addition to your basic plot. Ultimately, you can use some fun add-ons like Animate, where you make a static plot, and then let's say one variable is changing here, for example, here, and then you get a very cool GIF. So this is, by the way, this was generated with Gapminder dataset, and if you want to make this exact plot, you just come out of the Genomics folder, go into the Gapminder folder, and you'll see the code for this. Okay. So here, I'm also showing you very briefly. So here, basically, I'm taking the data. I'm specifying what are the variables that I'm plotting. I'm saying the size is by population, and the color is by country. Okay, and the facet, it means each block. I'm separating it by continent, so I don't have them all together. There are already way too many data points here, right? I'm specifying that I'm using a log scale. And then I can customize every single step. That's what I'm showing. Again, don't worry that there are too many lines. We'll do it step by step as always. But this is to say that you can make fancy plots, and very easily too. And then all I have to do is take the exact same code as before and add these two lines. Actually, it's just this one line here, because this, I'm just adding title to the plot. I'm just saying the transition time is here, and that's it. It becomes an animated plot. It's as simple as that in Ggplot. And I'm saying it's a linear scale. And finally, we'll close. I've already shown you right_delim a couple of times, right_tsv. But in addition to saving your data, you can also save your plots by using ggsave. And with this, we'll go back to RStudio. All right, part 4. So now we'll actually be recapping quite a few things that we learned using Tidier and dplyr, because you need to do a few different things to your data and bring it to a suitable format before you actually do the visualization. Okay, so let's first start here with this first chunk. And we are going to make a very simple bar plot. Let's give some room, because your plots are going to appear here. I'm just going to give this some room so that you can see it. So if you notice here, I'm taking the gene look. I'm not taking the entire data set, because for this particular set of plots, I'm going to use only my gene location information. Okay. One other difference that I want to point out is that we use pipes when we are going across functions. We use plus if we are adding different layers within ggplot. So you will see a lot of pipes and you'll see a lot of pluses. The pluses are indicating we are still within the visualization chunk of your code. Okay. All right. So I said we need 3 elements, right? You need your data. Check. You need your aesthetics. I'm telling what is the X variable here. It's my chromosome column. Check. The third thing is a geometry. I am saying I want a bar plot. And let's see what that looks like. That's it. You have a bar plot. Okay. Now it's not such an appealing bar plot. Look at this, right? You have overlapping characters. Maybe if I drag it, it'll be slightly better. But still, I wouldn't call it a nice looking plot. Now what can we do? Now we know how to make a plot. The next thing is, how do I customize? So here what I'm doing is converting these to factors. First thing, I told you, right? You have chromosome 1, then you have chromosome 10, 11, 12. I don't like that. So I'm just saying that I'm specifying the order in which I want these variables to appear. That's my first step that I want to fix. So here, these are already in factors. But what I'm saying is I'm specifying the order in which these levels happen. So I'm saying take this column, chromosome, and then I'm taking the chromosome. Then I go from 1 to 22, then X, Y, and M. And I'm pasting these together. So just run this part alone and see what that looks like, right? It basically gives me the string. So instead of typing these 25 variables, what I'm doing is I'm using a shortcut. I'm saying paste chromosome. That's the common part. Then I'm saying paste the next element, which is which chromosome it is. It can either be the number 1 to 22 or X or Y or M. And then I just say don't introduce space or anything else in between. Just directly paste them with nothing in between. That's the separator. And I'm saying this is the new order that I want. I run this. And now let's run this again and see if that is any better. Oh yes, it's better. I mean, it's partially better. At the least, you know that it goes from 1 to 23, then you have your X, Y, and M. Still, it's not that readable. I'm still not happy. Again, similar to your data frames, by the way, you can save your plots at any point. So for instance, if I run this, it's not going to print anything to the screen. But if I call that variable, similar to pulling up my data frame, it's going to pull up my plot. So you can save your plots, even the temporary ones that you make. I see that Sophia has a question. Give me just a second. I'll get to you. But as I said, I want to read what is here, right? Let's try a very simple thing. I'm just going to flip the coordinates. Maybe that will help. Excellent. That is all that I needed to do. Right? Because I just have these 3 numbers. I don't need a lot of space for that. And I have more vertical room. So this is much cleaner already. And by default, all Ggplot ones come with this gray background. You might have seen this and you find it annoying. So all you have to do is use the theme minimal, and then it gives you a basic black and white kind of thing. So there are several themes that you can use. And by the way, you may have noticed here, because I'm saving this in plot chromosome number genes, I don't have to copy paste this piece of code each time. I can just say this, and because this is a plot kind of class for this particular variable, I can just use pluses and keep adding layers or add modifications. See, I'm just starting with this and I'm flipping the coordinate. Or here, I'm just adding the title. I'm saying give the plot a title. And the X and Y labels. It's as simple as that. I just say plus, add the labels, plus, switch it to a minimal theme and flip the coordinates, and it can be in any order. Yes, Sophia. Side-by-side bar plots with error bars from a data frame, can we do that here? We might not have replicate data, and usually you need replicate data to get these error bars. Yes, we can do it, and I can point you to some examples, but we might not be doing it today. Simply because we don't have the right kind of data here to start with. Okay. So what are we doing next? Next, we want to take the length and we actually want to plot a histogram. This is a different kind of plot. So I want to look at the length distribution. So most of the gene lengths are somewhere in the middle, which is about 1,000 amino acids. No, actually, this is the nucleotide sequence, 1,000 base pairs. Very few towards the extremes. So this is generally to get an idea as to what is the distribution. But this is across all chromosomes. So you don't know what is happening still for one chromosome versus the other. Right? So what I'm doing next is I'm going to take the same plot, But now, by the way, did you notice, in addition to adding histogram, because it would have looked really messy, let's show you the messy plot. Because it's not on a log scale, this is how your plot looks. Most of your thing is an empty space. So all I did is basically add the log 10 here, just to make better use of my space and look at the distribution properly. And for most things, even today, if I need to make a brand new plot that I've not made before, I am going to Google it. Google is your friend. Whether it's for RStudio, Tidyverse visualization, just say R, Tidyverse or dplyr or Ggplot, your question. I'm pretty sure there are answers there, because tons of people have encountered the same issues and errors that you have. So there are solutions. And luckily, the R community is extremely helpful. The R ladies community, rest of the R community, they're all extremely helpful. So please look for questions. You will not be discouraged. Janal, I'll get to your question in one more plot. Okay. So next, what I'm doing is, as I said, this is giving me the distribution for across all chromosomes, but I actually want to know what is happening chromosome-wise. And I showed you in the Oceania other plot, I'm using something called facet wrap, and I'm saying by chromosome. Do you want to see what this does? The first time I was making these plots with Gg, oh, what is your problem? Let's see. The good thing is, we have our HTML. If something doesn't work, we can go there for our proof that something is working. Let's quickly go there instead of waiting. So this is how it would look. Again, it's not readable, it's ugly, all of that, yes, yes, and yes. But in just a snap, because of the facet wrap, you can now just get all 25. You get a 5x5 matrix of plots, one per chromosome, and you get the distribution. This is really messing with my space. And anyway, I know which chromosome it is, so I don't need this legend color here. So I'm saying remove that. I'm saying legend position equals none. And I get this. Really cool, huh? Just a couple of lines that I'm adding to this previous plot. And that boring plot becomes this. So this is really amazing. So if there is one suite of packages you learn for basic data analysis and visualization, I would say that is Tidyverse. Because it's so powerful, you can make so many cool plots. What is happening? Okay, something is supposed to be happening. A lot of data points, that's why. Okay, so this is a scatter plot with GM point. So what I'm trying to, let's see what I'm trying to plot here, right? There are two variables and I'm directly plotting the difference. How cool is that? I don't even have to pre-compute the difference and then call that new column. I can just say plot the difference, which is n minus start. And that's my x-axis. And my y-axis is my length. As you saw, the length was the same, regardless of what the transcripts were or what the isoforms were. And that's why you're actually seeing a distribution here. But I want to see how are they lining up. Right? So I have my length and I have my n minus start. And now I'm seeing this. This is good, but it can be better. First, it's not using the entire space. I don't like that. It's not a good plot when you really have to squint at it to find a pattern. So first, I want to increase the transparency. I want to adjust the size. I want to do a few basic things to make it slightly better. Now let's see how that helps us. And if that takes long, we always have our HTML. So the first thing is you don't have the dark blob. You are increasing the transparency with alpha, adjusting the size, the color, and everything. By the way, color is for the outside, fill is for what goes inside, and you can have two different colors for that. But then I say that's not very helpful either. I actually want this on the log scale because I want to see this line. Right? The length is almost always the same or shorter than the end minus start. Anyone want to guess why this is? Why the end minus start is kind of the upper limit and why the gene length is always lower? No? No one from biology taking a guess here? This is going back to our isoform thing, right? Your start minus end has everything. It has your introns, it has the entire thing. But your length is actually your final gene length. So you can, and you have these different transcripts, different isoforms. So almost always for any given point, you will see that the gene length is either the same or it's less than end minus start. End minus start being the absolute limits. And that is a really cool pattern that you can see by plotting your data and on the log scale. You want to add a couple of lines just for reference. You can also add that. And finally, this is just the 45-degree line that I'm adding to see is all the data prior to that, and yes, it is. So here I'm calculating the mean length and the number of genes, and then I'm passing that to ggplot and ggm. So these are basically I'm plotting the mean length and the number of genes with that mean length. Did you get the warning? Most likely I did get the warning, but I didn't run it here. So usually warnings are okay. Okay, that's a very terrible thing to say. Errors are bad, which means it's not even going to run. Warnings are telling you that something about the data is not sitting well with the expected outcome. So it will tell you if something is not the right format, there are NAs or something like this is happening. But usually it will still plot. So warnings are in some sense more acceptable. Okay, but now I want to know what is happening here. So I'm loading this other package called Ggreppl. If you don't have Ggreppl, then you'll probably have to install it. I hope I have it. Oh, I do. And this basically adds, everything else is the same as before. I'm adding a cline. See, there's a light blue and the gray. And then I'm basically adding which chromosome it is from. And Ggreppl basically says, don't put all, just because the points are close by, don't make all the labels overlap and unreadable. Just slightly stagger it so that it's easy to read. It's a very helpful package, especially for labels. And then you have this. Hedia, you'll have to tell me if I'm going over time. We are at 2.5 hours now, right? We still have half an hour?
Yes.
Okay, all right. So next what we are going to do is we are going to make a few box plots and violin plots. And why are we going to make them? It's because we're going to look at counts and we want to look at the distribution of these counts. And that's why we are going to look at the distribution with violin and box plots. Oh, gene log CPM. So you see this error here? It's a simple thing actually. It just says log CPM not found. And that's because I cheated. There were a few things here that I should have run, but I said, go take your break, and I didn't run any of these. So as I said, I'm calculating my counts per million here. I'm calculating my log counts per million, and I'm saving that. You want to see what I've done so far? This is how my log CPM looks. And this is my cleaned up version that I'm taking. In fact, I've done one more thing here. Remember, it was just A and F. That was not super helpful. So I basically added one more thing. I just said if it is A, please make it adult, otherwise make it fetus, because that's easy for me to read, especially when I'm plotting. I don't want to be dealing with characters, but I want the full names spelled out. Okay, so that's all that I'm doing here. And now I'm just plotting the gene log CPM here. Something is coming. Wait a minute. Ooh, wow. Okay. So here, there are a few things to look at. Let's plot this. So here I'm saying, I'm specifying what my X and Y is. But you will see that it's flipped because I'm using coord flip that we used, remember, for the bar plot as well. That's why my X and Y are now flipped. But this is basically my sample and my log CPM flipped. I'm saying, please color it by the tissue. So now, and then I'm saying the adult is the full line and fetus is the dashed line. So these pairs will basically have the same color because they are both stomach. One is adult, one is fetus. Okay. This is also stomach. This is a different technology, but it still gets the pink and the full line. Anyway, so this is the guide to how to read this box plot. And these are the different elements that we have. And I've set T minimal, so it just has the white background instead of the gray one. And it has the outliers. It has the outliers by default. And now I'm converting it from a box plot to a violin plot here. Violin plots are only slightly different from your box plots in that they kind of show you the shape of the distribution as well. I mean, you're pretty much using the same outliers and the median and the interquartile ranges, but you also have that slightly extra piece of information. Okay. And if you want your outlier data to look slightly different in your box plot, you can change the transparency and the size of your outliers. So these are all different parameters. You can keep customizing. That's the thing with Ggplot. It's a blessing and a curse. You can make it as beautiful as you want, but at the same time, you can spend hours and hours and hours fine-tuning your plot until someone says that's enough. Just give me a plot. So here we have changed a few things. The scale, and we have basically fixed the outliers slightly. Because in the first plot, when we made the box plot, these did not have the transparency. Now all of these these are going to take slightly longer time. So I'm just going to scroll on quickly to show you what's happening. See, this is how it looked in the original plot, right? Tons and tons of outliers. I reduced the size. I reduced the alpha. Now it is much, much more readable. Then what I'm doing here is basically, I'm only selecting these colon samples, the AF colon, or it should be the BF colon, basically the fetal colon sample, right? And then I'm selecting only the log CPM, the sample, and the gene ID. And then I'm making a scatter plot, which is a geom point. And I'm going to quickly go here to see this is how it looks. So first, I'm very glad to see that regardless of where the sample is coming from, meaning the technology, first, I'm happy to see that they're correlating, right? These are both fetal colon samples. And if there was no correlation and it was all over the place, it would be a little upsetting. How do I trust my samples or my data, right? So this is a good basic check to make sure that your data is still kind of making sense. These are a few things you can do in addition to the summaries that you did. Visual checks to make sure that everything is still on track, looking fine, you can proceed. So what I'm basically doing is taking these variables, subsetting, and I'm doing all of it in one go, right? So these are all from dplyr, and then I'm going directly into ggplot, and I'm saving the plot. Some data sleuthing. Okay, so what we are going to do now is look at, I won't tell you what I'm going to look at. Let's see if it's helpful for you to tell me. So I'm taking the log CPM data again. Okay, I'm only taking the unique gene IDs and I'm just getting the total number of genes. Okay, you want to see what the total number of genes are? Oh, the same basically, 57,000. And then I'm only looking at the high variance genes. So I'm grouping by gene ID. I get the interquartile range. And then I'm taking the top ones here and saying I only want the ones with a high variance. You want to see how many there are? How many high variance genes there are? That's a manageable number, right? We started from 57,000. We only have 2,800 genes that are in this range. Now, I'm going to restrict this further. I'm saying I'm only interested in genes that are from chromosome Y. And see what I'm doing here. This is the ampersand, right? So it means check for two conditions and both the conditions have to be true. So this is my original data set, which I've subsetted with only the location information, right? But these values are from the gene log CPM, which is the other part of the data set. You see what I'm doing? I had the full data set. I split it into two. One with the sample counts and everything that I converted to log CPM. This one I kept intact that has the gene location information. What am I doing now? I go to the log CPM data, find the highly varying genes, right? I subset. Now I know which are the genes. I can come back to this first one, which has the location information, and I pull out only those rows that are in this high variance gene list. And how do I do it? It's actually fairly simple. I say the gene IDs have to be in this list. Where am I getting the list from? Whatever I did up here. It's as simple as that. I'm adding one more condition though. Don't give me everything that's there because it's 2,800. Only give me ones that are from chromosome Y. And then what pull does is it only pulls that one column. So if you do this entire thing, it's going to give you everything from gene log that has chromosome Y with the highly varying ones, right? But I'm saying once I use pull, it will pull only that column or those gene IDs it's going to pull. Okay?
Sorry,
Then
what does
sorry.
yeah, what does pull do? Because I couldn't understand it.
So pull basically gets me the gene IDs that correspond to this condition that I'm applying.
Okay.
It is a slightly different variation of the select. Select will give you the whole column.
Yeah.
Let's see the difference between that and select right here. So select gives you this way in a column, right? Because you're selecting the column. Pull basically pulls the values from that column that correspond to that condition.
Thank you.
So you can give this you can pass this vector to your subsequent function that you're doing. So basically here, I just want the names of those genes. That's why I'm using pull instead of a column select variable. Okay?
Thank you
next,
very much.
what I'm doing is, then I'm going to this log CPM. And now again, I'm filtering. I'm going back to the other one, by the way. Okay? Now I know that I want which one has chromosome Y. That's why I had to go to the other one, right? Because my log CPM data did not have my chromosome name. I know I'm asking you to remember a lot of things, but let's look at it. So this is how my gene lock looks. Oh, there is a head here. Let's go there. So this is how my gene lock looks. It has the chromosome information and the gene ID, but no log CPM. My log CPM has my sample information and the log CPM, but it does not have the chromosome information. But I want highly varying genes that are from chromosome Y. That's why I'm going back and forth between these two data frames, which, by the way, we got from the same big original data frame. Okay, so now I have this chromosome Y, high variance ones. I'm going back to log CPM, and I'm doing the same thing. Only pull once. Only pull those values. Oh, something was not right. Okay, I hadn't saved it in that variable. That was the error. Okay, so now it's only pulling these values for me, which is the 238. Next, time to plot. Why did we do all this hard work? So what I'm doing is basically the same two lines, filter, but I'm adding Ggplot. And I'm doing a box plot here. You know, it has no legend, it has been flipped, you know all of these now. Let's make room for the plot. Okay. So this is the log CPM, which is basically, you can think of it as how well expressed these genes are. If it is high, it means it's highly expressed. If not, if it's close to 0, it means those genes are not expressed. Okay? So you're kind of seeing, in some sense, a bimodal kind of pattern, right? You're seeing these samples that have fairly high, because it's a log scale, right? You have fairly high non-zero values, and these are all close to 0, even on the log scale. So what do you think this means? Any guesses on what these samples could? By the way, this is from chromosome Y. These are chromosome Y genes with these values. Any guesses on what is different between these samples versus these samples? No? It turns out that these are actually samples that were derived from female patients. So the chromosome Y genes are not expressed. And you didn't have to know that metadata. You didn't know that beforehand, right? You got all of this from the analysis that we just did. We are able to infer.
Sorry, you have outliers. This is like incorrect outliers.
Yeah, I will get to that, because
Okay,
these
thank you.
were these were actually mixed samples. So in these samples, they had I think 2 or 3 females and 1 male, and these other ones were primarily male samples. That's an excellent catch, Ali.
Okay, thank you.
But the beautiful thing is, you can actually figure this out just by looking at the data. You don't need to go back to NCBI, pull up all the metadata, read in the paper, where did they get the samples from? You can see it for yourself with the data that you have at hand. This is really cool, right? And finally, with that, you can go back to saving the plots that you made so far. So you've made a bunch of plots, some that you're happy with. You add whatever title you want, label name, and then we come to this chunk where you save it. So as I said, write underscore TSV is going to be for saving it, or write underscore CSV or DLIM, and GG save is going to basically save your plot. Well, let's try GG save, because I don't seem to have a working example. So what's the file name? I want to say go to Genomics and chromosome Y high variance plot. Then which is the plot that I want to save? I want to save this one. So I didn't run it. So this is not going to work, but let's still do it. What are the other variables? I want to save it as a PNG. What else do we want to specify? I want a high quality plot for my publication. So I say DPI 600. And I run it, it's going to give me an error. Why? Because I didn't run the previous one properly. So let's run this.
We have a question. So the question is, what is the advantage of saving with GG save instead of just exporting the figure?
By exporting the figure, do you mean from here? The advantage is, first, I'm doing it programmatically. So when I'm running it for a whole bunch of files, it'll automatically save it. I don't need to look at the plot and save it each time. And I can specify all these parameters rather than, okay, let's do what you're saying. Let me finish this. I'm going to, now that I have this variable, I'm going to rerun. And hopefully it has saved it. There you go. It has saved it as a PNG. Okay. Now this was easy enough to do, right? And I can do this because I have it in my code. I can do it for any variable, any number of variables, save all of them in one shot, put them in a folder, and not look at it until I have to look at my plots again. But if I'm doing it here, I will just go to export. I'll say save as image. I have to specify here. I have to specify all of these. And I still cannot specify my DPI very clearly. And each time it's not going to remember the file name or any of this, because I can resize it and it's going to look different. And it's not even working, which is perfect for our demo. Okay. Right, but I can, it gives me just the flexibility and the reproducible part by just putting it in the code. Let me just say that. And sometimes it also takes a here, it's okay. We subsetted, it's only chromosome Y. We didn't have a lot of data to work with. But as you saw, it also takes a very long time to visualize each of these. So instead, if I'm directly saving it, sometimes it's helpful. And the other advantage is, now let's say you're working on your server or your HPC, right? You might not really have your GUI version of it, your RStudio thing, but you might want to run it on tons and tons of data sets. So you can try whether your things are working properly and they look fine when you have 1 or 2 examples locally. But when you're running it on a large scale, it makes sense to put it in a code and just get the outputs. Later, you can push it to GitHub or something and then just look at the plots alone locally. Because you don't have the option of GUI-based export when you're working on, say, HPC or the server. All right. So you can save your plots, save your data, and do all of that. Which brings us to, these are all the things that you learned today. How about that? Install packages and library, you probably already knew, but you know how to import data, look at your data, get a quick snapshot, basic important functions in tidier and deployer. Ggplot, again, just the 101 version of Ggplot, because there's lots more. Ggplot alone, we can do a series for 4-5 months at least. And then there is the Ggplot extension gallery. And then finally, you know how to save all of your work. So this material, especially for the genomic data, was developed by Arjun Krishnan for one of his courses. And I adapted a lot of this for the workshop. He's also an assistant professor at Michigan State University, and incidentally, also my husband. So a lot of this work was done together with him. And I would also like to acknowledge, let me see if I can go back to my slides. So after covering all of this, I will recommend that you definitely look upon these amazing books. All of them have free online versions. R for data science, hands-on programming with R. And if you want to add more statistical analysis, take a look at Daniel Navarro's book too. And few things, if you want to learn more about pipe or the distill theme that I use to generate my R markdown, you can also look at that here. So with that, I would like to thank Arjun, Our Ladies East Lansing, where I had a chance to kind of fine-tune this presentation by giving them a few presentations in the past couple of years. The Our Ladies global community, of course. It's been amazing interacting with them. And with that, I'm happy to take any questions.
Thank you, Janani, for this amazing workshop. So please, if you have any question, you can unmute yourself or put it in the chat.
Yeah. Yes, Olu of me.
Okay, thank you, Ma, for a wonderful presentation. I want to ask if, for instance, I develop a visualization with Ggplot too. Is it possible that if I want to overlay like 3 layers on the same plot, is it possible that after I put the plus sign, I specify the aesthetics for the second plot? Will it work that way?
Yes, you can keep adding aesthetics to the same plot. You can add different layers to the same plot. For instance, when I added those ab lines, or when I added the linear model with the shading, or I added labels, you just do plus, 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 and you keep adding all of these layers. And it can be done very easily.
Okay, thank you, ma'am.
You can call me Janani. You don't have to call me ma'am, but thank you.
Any other question?
I'm glad everyone found it helpful. You can start this repository if Tidyverse changes, if these functions change. I'm going to keep this updated as much as possible. So you can go to the repository and just start it here, or fork it if you want a local copy that you want to work with.
So Jawed is asking, I don't know about genomics. Where can When can you know about data?
So there is more information in the Markdown and the HTML that I gave you where I described the samples. But as you said, if you're not familiar with genomics at all and biology, I would say you can go to this other folder called Gapminder. And you can basically look at that HTML. And that's going to be a different data set altogether. That's going to talk about your GDP and life expectancy and things that make a lot more sense than genomic data sets. So the HTML and the PowerPoint for that is also available. Let's see. So the structure of the workshop is going to be very similar, but the plots, the summarizations and everything are going to be different. So also if you're new to, if you're new to Tidyverse and you want to try it with multiple data sets, even for those who are from biology, I will recommend you look at the other one. Because you'll see slight variations, different regex that you're using. And the plots here are also going to be different. So these are the ones that I also showed you in the presentation. Oh yes, Ocon, I do remember. I will stay on once we conclude. Okay. I am glad everyone found it useful. I hope those with biology or without biology, both of you benefited from it. That's why I kind of have two versions here.
Franklin is asking, Tidyverse is recommendable to big data?
Big data, yes, very big. I would recommend data table. Data table is much, much faster. And especially when we are rendering things on our Shiny app, for example, for all of that, we use data table.
Yeah, and with data table, you can go from data table to data frame and then vice versa. So it's really good.
Yes. And it's much faster too. So for example, let me show you an example here where we use data table. Right. So this is basically a web application or a Shiny web app that we developed. And here, it basically has a lot of data. So if for it to be dynamic and fast and to be able to subset and do all the visualization and everything, for this application, there is no way we could have done it with only Tidyverse. So for this, we use actually data table. So for all the summarization and visualization that we do in this app, it's much, much faster. It makes a huge difference. Lucas, you have a good point. What is big? For me, what I consider big is if Tidyverse crashes or my RStudio crashes, I go very quickly to go running to data table. But I'm sure there's a better definition of what is big.
Yeah, it's same for me.
Any other questions?
When it puts
usually
you can assign a vector of 25 giga or something, so it's big.
So that's what happens, right? If you're comfortable in your space, you're doing something for a long time, you just do it. When something breaks and it stops working, then like, okay, time to look for a new package that will help you with larger data sets.
Yes.
Data table. And you can look for DT and data table. DT also works very well with Shiny. I will type it out.
Excuse me, just I have a question about the data table package. Does it work with R version 4 and more, or just because just I got like an error message? No, but just I got like an error message. It was built under R version 4.0, just 0.0.5.
So
maybe
your question,
just some objects inside just this package, I think so.
so you're saying that data table does not work with your R version? Is that what you're saying?
Sorry, just some objects under this package, I think so. Like, I don't know, it might like be an error message. Okay, never mind. Thank you.
Okay. I'll have to check the specific things. Unfortunately, I don't have a blanket answer for you. But data.table is one thing that I use, and the other thing that works. This is a JavaScript wrapper of data tables that works very well with rmarkdown, flexboard.
Is for what, Femi?
Shiny. Sorry.
Your mic is on.
Hello.
Yes.
Yes, we are hearing you.
You promised that at the end of the lecture you help me out with the installations for Tidyverse.
Yes, I
will.
will.
All right. Thank you.
Okay. Any more questions before we help fix Tidyverse?
I think there are no more questions. So we want to share. Monet will share her screen about our next meetup. Monet, can you share your screen? So thank you very much, everyone, for joining our workshop. And thank you, Janani.
Yes, for this incredible workshop. We have learned a lot from you today.
My pleasure. Thank you for inviting me.
Yeah, it was a great session. So our next R for Bioinfo will be about omics integration and supervised machine learning. So it will be in two parts. We will have the first part on May 8th and then the second part on May 22. So please join us.
I will share with you the meetup link so you are able to fill out the Google form and to register.
Yeah, it's a very interesting subject. Thank you.
Sounds awesome.
See you next time, and we'll be happy to share you all the workshop materials later, the recording video, etc.
Thank you. Thank you.
Thank you all.
Thank you so much for organizing this stuff. Thank you so much.
Thank you.
It's
Thank
helpful.
you. You are
Thank you.
welcome.
Thank you.
Thank you so much.
Thank you. Bye-bye.
Bye.
Bye.
Thank you.